Hey everyone, here is your first video about genetics. And in this video, we're just going to talk about some of the vocabulary words that we will use when we talk about genetics. So let's get started. The first thing we will talk about is Gregor Mendel. Now, he is considered to be the father of modern genetics because he was the first scientist who really started thinking about how different traits are inherited from one generation to the next. He was actually, he has an interesting backstory. So he was, as a child, very poor. He didn't always know where his next meal was coming from, but he was really interested in science. And one way for him to become a scientist without having to pay a lot of money to go to college, because his family did not have a lot of money, was for him to become a monk. And he did. He became a friar or a monk, a religious man, and he worked with pea plants when he lived in a monastery. And this was around the 1860s. Nobody knew what genes were. Nobody knew what DNA was or the structure of DNA. We just thought, hey, parents, maybe they pass some characteristics on to their children, but do we know? Well, he called them factors, the things that we pass on to our children, and now we call those alleles. So that's a vocab word that you'll see, alleles, and that's like kind of the genetic material that gets passed on. So he looked at pea plants and he crossed a purebred round pea plant with a purebred wrinkly pea plant. And all of the children or offspring of those pea plants were round. And he was like, well, hang on. What happened to the wrinkly trait? Like, how did that just disappear? Well, then he crossed a one from that generation that had half the genes from a round one and half from a wrinkly with another one just like it. And he found that of those offspring, there were 3 out of 4 that would be round, and then 1 out of 4 that was wrinkly. And he was like, well, that's interesting because that one's wrinkly, and that looks like the one that was kind of basically like its grandparent. So, where did that trait come from? Well, it got passed down somehow. And what we found out nowadays is that although these ones look round, they actually carry the wrinkly trait, and that's marked with that little s there. And then that's how they could have offspring that are wrinkly. So let's get a little bit more details with vocab for genetics. So first thing you really need to know the difference between is a genotype and a phenotype. So a genotype is the set of genes that an organism carries, and it's written as alleles, and we use uppercase and lowercase letters to mark our alleles. So we use like an uppercase B to mark a dominant allele and a lowercase b to mark a recessive allele. And we'll talk about those words in a second. But an example of the alleles that you might have would be like big B, big B, little b, or little b, little b. All right, now we got phenotype, and that's the actual observable characteristics, like how something looks or what it's able to do. Like the fact that I can curl my tongue into like that U shape, curled tongue, is an ability that I have. That's a phenotype that I have. And actually, interestingly enough, my grandmother can't do that. She doesn't have that phenotype. So the phenotype is your ability or how you look, like what color your eyes are, what color your hair is. Can you curl your tongue? Do you have double jointed fingers? Like all these different things that you have that are your traits, those are your phenotypes. But the actual set of alleles that you inherited from your biological parents are your genotypes. So you heard me mention dominant and recessive. And what that means, well, dominant means that the trait is usually expressed most often. If you even have one copy of the dominant allele, then you are going to show up as a dominant phenotype. So, for example, the allele for tall plants is big T. So, if the dominant allele for is big T, then the recessive allele is little t. So, if a plant has a genotype of big T, which means it has 2 dominant alleles, it's going to have the dominant trait. And if it has the genotype of big T, little t, even though it has that recessive little t, it still has that dominant big T, and it's going to appear to have the dominant trait or to be tall. It's gonna be a tall plant because it would have at least one copy of the dominant allele. So whether you have one copy or two copies of the dominant allele, that is going to show the dominant trait. Now, the recessive trait, if you think back to Gregor Mendel, actually the recessive trait here would have been to be wrinkly as a pea plant. And that trait is recessive because here, when you had big S, little s, the big S was the dominant allele and that won out. That's why this one's round. The little s is there, it's carried, but it's carried as a recessive trait. And in order to show the recessive trait, if you look down here, to show wrinkled, or even up here to show wrinkled, you have to have both recessive alleles. So the recessive trait is only expressed if there is not a dominant allele present. An organism can have the recessive allele without showing it, just like we saw with the pea plant. Next up, I have homozygous and heterozygous. So homozygous, you see the word homo in there, and that means same. And so you see two of the same alleles would be considered homozygous. And you can be homozygous dominant, like big T, or you can be homozygous recessive, which would be like little t. And if you are homozygous, you are known as a purebred individual. So if you're homozygous, if you have a plant that has two big T's, it's going to be homozygous dominant for the trait for being tall. And if you have a plant that has little t, those are both the same. So it's going to be homozygous recessive, and it would appear as the phenotype to be short. For the other vocab word here, heterozygous, that means an organism that has two different alleles. And when you have two different alleles, if it's Mendel's genetics, it always shows the dominant phenotype. This is known as a hybrid. So two different things, just like hybrid students are doing two different things. They do some work at school and some work at home. The hybrid individuals are heterozygous and they have two different alleles. So the genotype big T, little t is actually hybrid or heterozygous. And the phenotype, the way this would appear, would be tall. And the reason that it would be tall is because the heterozygous ones always show the dominant phenotype in Mendel's genetics. All right, next up, I have autosomal and sex-linked. You're going to see this with some traits that we're going to talk about in a couple videos. The autosomal means that genes are located on any chromosome other than your X or Y chromosome. So you have 23 pairs of chromosomes, and the autosomal chromosomes are numbers 1 through 22, pairs 1 through 22. And if you have an autosomal condition, then they can sometimes skip generations, and we can look at them and study them in what we call pedigrees, which we'll talk about. A sex-linked trait or a sex-linked gene is actually going to be located on the X chromosome or the Y chromosome, and those are the chromosomes that determine your gender at birth. So if it is on the X chromosome or Y chromosome, it is a sex-linked gene. And unfortunately for males, they are more likely to suffer from sex-linked disorders, which we will talk about in the next couple videos. All right, there's two laws that you need to know when working with genetics, and those are the law of independent assortment and the law of segregation. The law of independent assortment basically means that when you get your genes, just because you have a gene for a certain trait doesn't automatically mean that you're going to get another gene that you might have. So just because if you make offspring, just because you are passing on a trait of having blonde hair to an offspring does not automatically mean that you will also pass on blue eyes. They are separate genes and they are passed on independently of each other, separately of each other. Another example would be just because I can curl my tongue does not mean that I am double jointed. the gene for curling tongue and being double jointed are separate genes, and they're passed on to offspring independently of each other. The reason for that law of independent assortment is actually, if you think back to when we talked about cell cycles, it's because of that crossing over that takes place during prophase 1 of meiosis. Next up, we have the law of segregation. The law of segregation is that for any trait, each parent's pair of alleles is going to be split, and only one gene will be passed from each parent to an offspring, and it's up to chance which gene will be passed. So for law of segregation, look back to Mendel's pea plants. See how this pea plant has a big S and a little s? It's completely up to chance with whether it will pass the big S or the little s onto each individual offspring. So that those S's will actually be split during meiosis, and it's completely up to chance whether any random sperm or egg cell would receive the big S or whether it would receive the small s. So that is the law of segregation, just that one gene will be passed from each parent to an offspring, and it's up to chance which gene the parents pass. The last vocab word to think about is punnett squares, and we will talk about punnett squares in the next video, but you might have seen these types of squares before with letters in them, and they help us show the possible combinations of genes for any offspring of two parents. Thanks for listening. We're going to look at punnett squares next.